in Kelton this week, and Chris is gone again. Most of what we did for the finding our answers all came from the notes from our videos and the lecture notes in the PowerPoint. So we'll go through. We'll start with number one. The questions they concern molecules involved in translation. These weren't too bad to find. They're mostly all the molecules and factors that go into involved in translation. So we have indicate the groups of enzymes that covalently link amino acids to tRNA. That's the charging tRNA and the initiation tRNA. 4 proteins that consume GTP is the EFTU factor, the GFG factor, the IF3 factor, and IF2 factor. 1 molecule that functions as a ribosome during translation is the 30S subunit. 2 proteins that recognize stop codons are the release factor 1 and release factor 2. 1 molecule that forms a hydrogen bond is the 16S RNA of the 30S subunit of the Shine-Delargano sequence. Here's number 2.
So number 2 is the first amino acid, the prokaryotic proteins, is the N-terminus. Is that how you say it?
Sure.
Hopefully, yeah. And then an E. coli protein called elongation factor, translation. What's the U stand for?
I can't remember the U.
I don't remember the U either. But yeah, that's the answer for that one. That delivers the charged tRNAs to the A site of the ribosome during translation elongation. And then the initiator tRNA binds to the P site of the ribosome, whereas the remaining charged tRNAs bind to the A site of the ribosome. And then the EFG is the translation factor that helps the ribosome move 1 codon to the 3 direction along the mRNA. And then the IF3 is a translation factor that guides the mRNA to the 30S ribosome subunit. And then the 5-AUG3 codon is the start codon, while the UAA is an example of a stop codon. And then G-generate is a term that indicates that more than 1 codon can specify the same amino acid. So throughout our notes in the PowerPoint, it showed like a picture of more than 1 possibility of how each one could have a different letter thrown in there. So there's more than 1 possibility. And then the F, or the final amino acid, is located at the carboxyl terminus of the polypeptide.
So number 3, this has to deal with the wobble codons. So from our understanding, as long this question's asking for the anticodon, so for the U's you would have A's, and then with the wobble, the third letter could be any of the 4. So each one of these we would go through. Instead of doing 5', 3' every time, we just listed them all 5' to 3'. So for the first one, UUG would also be indicated as AAU, AAC, AAG, and AAA. As long as the first 2 are the anticodons of the UU, the third one doesn't matter. We did that same thing for all of these. For example, on C, the UAC could also be UAG, which is a start codon, so that one might get canceled out. It also would be UAA, UAU, and UAC. And then E, we have CGU, CGA, CGG, and CGC. And those would be, as long as the first 2 are the same, the third one doesn't really matter. And then number 4, we had to do a little bit of research to figure this out.
So number 4 was basically just some math problems, and we hopefully did this right. We'll see if we did. But it says that the average amino acid is 125 Daltons. So then we did 218 kilodaltons times 1,000, which would give you 218,000. Divide that by 125 to get 1,744 amino acids. And then from there, it is the 3 nucleotides to form a start codon And the example is the 5 AUG to the 3, and that's the length of it. And then C is asking for how many GTP molecules are consumed in synthesizing each copy of protein X. So there are 2 GTP for every amino acid in initiation and 2 for elongation. And then there's also 1 GTP for terminating, which gives us 4. So then we go 4 times 1744 plus that 1 equals 6,977 GTP to synthesize 1 copy. And then D, we weren't really sure on this one, and so we left it blank. We just didn't really know in the notes and PowerPoint where to find it or for sure what the answer was. But for E, we found out that it was charging tRNAs.
Nice. For number 5, these yellow highlights, they just mean that I used them somewhere else so I could keep track of what I was using. So for A, which of the above is involved in translation initiation? You have the initiation factor 3, initiation factor 2, the tRNA FEMT, and then the 5' AUG is a start codon, and the Shine de Larangro is also part of initiation. What's involved in elongation, you have the elongation factor TU, the 16S RNA, the EFG, the 23S RNA. Which of the above is involved in translation termination? The release factor 1, and then the 5' UGA is a stop codon. Which of the above is a component of the 70S ribosome? That's the 16S and the 23S. Those are both part of the 70S. Which of the above is found in the mRNA? The Shine de Larangro, AUG is a start codon, tRNA ferment, and the UGA is the stop codon. We couldn't find this lysyl tRNA synthetase. So we didn't use it because we didn't know what it was. And that's kind of what we got for the week. Hopefully that helps you guys find some stuff out.